Hello, everyone. Want to thank you for joining us for this month's Roundtable for the Innovative Drug Discovery and Development Consortium. Our host for today is Dr. Jen Wang, one of the newer members of our IDDD steering committee. Jen joins us from Baylor College of Medicine and has organized this first of our IND to concept series. And so we're really excited to get this day started. And with that, I will turn it over to Dr. Wang.
Yeah, thank you so much then for the introduction. So today is actually the first GCC IDDD roundtable for this academic year. And we have the topic as From Hate to Hero, the modern small molecule Odyssey. So we got two fantastic speakers, Dr. Adam Gilbert and Dr. Nicholas Meanwhile. So I'll first introduce Dr. Adam Gilbert, and then he will give a 30-minute talk. And after that, Dr. Meanwhile will give a talk, and then it will be the Q&A sessions. And you guys can actually ask questions in the Q&A box. So Dr. Adam Gilbert is currently a scientific advisor to Axiom Therapeutics, is on the scientific advisory board of foremost Trim Tech Therapeutics and Garo AI. And he was most recently an executive director and external accelerator lead at Pfizer, where he created a proactive partnership and business development deals to build value for Pfizer portfolios. He has more than 30 years of experience working in biopharma, and which included leading Pfizer's design and synthesis science platform, medicinal chemistry group, and successfully delivered targeted covalent inhibitors, which he will talk about today, and protein, homeostasis modulators, vaccines, you name it. He is a co-inventor of the JAK3 covalent inhibitor he's gonna discuss today. And also his group helped deliver preventive respiratory virus vaccine. And also the one you guys most familiar with is the COVID-19 mRNA vaccine. And Adam received his PhD in organic chemistry from Columbia University. Adam, take it
Yeah,
over.
thanks very much, Jen, and for the invitation to present today. I'm really honored to be able to present. So let me know if you guys can see these slides.
That works.
It's working? Okay, great. So today I'm going to give I guess, a short version of a talk that I gave at Baylor a few years ago on the discovery of ritlecitinib, which is a JAK3 tech kinase inhibitor that was approved for alopecia in 2023. We started out trying to design this to be a JAK3 inhibitor specifically. We got fortunate in that it turns out it inhibits JAK3 and a number of tech family kinases. But as an overview, what I'm going to talk about, I'm going to talk about the JAK pathway and cytokine signaling in general, why covalent inhibition of JAK3 is required to obtain selective JAK3 inhibition, and talk about how this covalent inhibition requires a really unique parameter optimization for medchem when we did this in sort of the early 2000 teens. And then the discovery of ritlecitinib, the commercial name is Litfulo, or PF 0665-1600. The disclaimer below in terms of all the studies on animals were done in accordance with regulations and established guidelines. So the JAK-STAT signaling pathway has been, it's very, very well known, and it has been targeted by sort of the pharmaceutical industry for many years. And there are many, many great products that are on the market that inhibit various facets of this pathway. Cytokines bind to cytokine receptors, and once they do that, they activate these JAK kinases that signal in pairs. And these JAK kinases then move on to phosphorylate STAT proteins, which tend to dimerize and move into the nucleus where they, facilitate gene transcription, and they lead to immune stimulation or inflammatory responses. And on the right-hand side, you can see sort of the pairings of how JAKs tend to signal and what cytokines they're impacted by. JAK3 is actually unique in that it only signals and partners with JAK1 and facilitates the signaling of these gamma chain cytokines and leading to phosphorylations of mainly STAT3 and STAT5. But sort of there are many, many drugs that are on the market that affect these biological targets. They elicit different STAT gene signatures, and you get different flavors of function by coming up with different ways to sort of impact the signaling pathways of these JAK-STAT pathways. So a little bit more about kind of JAK3. Again, JAK3 signals in conjunction with JAK1 and responds to the cytokines in the upper left. And JAK3, in conjunction with JAK1, is involved in the growth and maturation of lymphoid cells, differentiation and homeostasis of T cells and NK cells, B cell switching, and some general inflammation. And if one were able to sort of selectively inhibit this pathway, you potentially would have a very clean path towards therapies for a number of autoimmune indications, such as alopecia, IBD, rheumatoid arthritis. So you would have something that was very selective and potentially safer than inhibiting the other JAKs that are involved in kind of broader cytokine signaling. Although there are very, very good drugs that are on the market that do inhibit many of these JAK enzymes. So in terms of how one is going to think about building a selective JAK3 inhibitor, Pfizer had this drug that was approved actually quite a while ago, tofacitinib, which inhibits, it actually is sort of a broad JAK inhibitor. It inhibits JAK1, somewhat JAK3, TYK2, not so much JAK2, and it's approved for rheumatoid arthritis. There are some potential side effects with it, and you have to be careful how you dose it. So Pfizer had an interest sort of back in the late 2000 aughts, I would say early teens, to come up with various flavors of JAK inhibitors. And for JAK3, if you look at the ATP site, which is where most kinase drugs are designed, the sequence homology between JAK1, JAK2, JAK3, and TYK2, TYK2 is a JAK, even though it's not called JAK, they're very, very similar. There are some notable differences in H10 and C4. H10 was particularly attractive to us since in TYK2, JAK1, and JAK2, the amino acid residue is serine. In JAK3, it is cysteine. This is a dock structure of tofacitinib overlaid with JAK1, JAK2, and JAK3. This is the residue at H10, and you can see the methyl group in tofa points down directly at the serine cysteine cluster. And because these residues have different nucleophilic character, we originally envisioned a strategy where we would try to design a covalent inhibitor which would react with the cysteine of JAK3 that would not be reactive to the serines of the other JAK family members. So there are challenges in terms of thinking about trying to execute this strategy. One of the problems with JAK3 that we noticed is that there is biochemical inefficiency of inhibiting JAK3 compared to the other JAKs, and this has to do with the Km of ATP that JAK3 has. So if you look at the Km of ATPs of the various JAKs, JAK3 is particularly low. And if you put this Km in this Chang-Prusov equation, which can be useful for correlating what the shift in inhibition would be when you inhibit a kinase at the Km versus inhibiting it in sort of blood or physiological ATP, which is 1 millimolar, you can predict that there would be quite a large shift. This is the theoretical shift for all the JAK enzymes. The experimental shift tends to line up somewhat. So JAK3, you would predict there would be a 125-fold shift going from Km of 4 micromolar to a millimolar. In fact, it's about 100. You can see here we plotted, a number of JAK3 inhibitors that we developed for our program at the Km of ATP versus 1 millimolar ATP, same type of assay, and in general, there is this 100-fold shift. So if you usually run kinase programs where you're looking at things at the km of ATP, if you have something that's selective for JAK3, as soon as you go into a more physiological environment, a very selective inhibitor actually may not look so selective. And that is actually what happened when Pfizer was working on tofacitinib back in the 2000s. It was a self-described selective JAK3 inhibitor, but as they better understood the compound, it actually is more strongly inhibiting of JAK1. So this was our strategy, again, is to work in this pocket to target cysteine 909. Another thing that we had to worry about is that if you look across the kinome, there are 11 kinases in total that have a cysteine at residue H10, the same residue that JAK3 has the cysteine. If you look and think about would it be a good or a bad thing in order to inhibit some of these kinases, again, we're designing JAK3 inhibitors for autoimmune indications. Some of the tech kinases look okay, right, for autoimmune indications to inhibit. We wouldn't worry too much about BTK or tech, but you do have EGFR, HER2, HER4. We're going to stay away from those. So this was an additional challenge that we would have to think about. And if you think about just one more challenge, I'm not trying to make this dramatic in terms of all the challenges, but there were some things we had to notice. So there is a safety component about trying to stay away from some of these tech family kinases, but the thinking at the time is that one really wanted to have as low a dose of a covalent compound as possible. I don't think it's as much. I don't think people are worried about it now as they used to be, but there was a worry when we were doing this that promiscuous labeling of proteins with a reactive covalent inhibitor could elicit this inflammatory autoimmune response by this danger hypothesis. So you could be dosing patients in the clinic, and they would develop this kind of broad immune inflammatory response, and patients could get in trouble, and it might be hard to monitor and head off. So we wanted to keep the dose as low as possible, and something was working against us with having the dose be low is that JAK3 tends to turn over very fast. Its resynthesis rate is on the order of 3 to 4 hours. We measured this by a standard pulse chase experiment. So we knew we were going to have not only a potent compound, but a very low clearance compound in order to be able to dose this once a day. If you look at other proteins which have been the targets of covalent inhibitor programs, Fa was an inhibitor program in the past that many companies went after. BTK, there are many BTK inhibitors on the market. These proteins turn over very slowly. So you can have very spiky PK where you kind of blast the drug in, it can clear fast, but the amount of protein that, is inhibited stays down for quite a while because it comes back very, very slowly as it's resynthesized. JAK3 is not like that. It'll come back very fast. So in thinking about this and where we would have to be to get a low dose compound, say less than 100 mgs per day, where the IC50 would have to be, and this is the shifted IC50 that I was talking about, it's not the KM. And we knew that we were going to have to cover the target very well, potentially IC80 at C average concentrations. We were going to have something that was going to be potent and clear very, very slowly to have a shot of delivering an agent that we would know would cover the target in the clinic. So we begin. So we worked on multiple series for this program. I think roughly 5 or 6. One of the initial learnings that we had was that as we looked at profiling compounds and assays, in vitro JAK3 inhibition, we measured JAK3 inhibition in cells in PBMCs. We measured JAK3 inhibition in whole blood, which is actually the physiologically relevant media that would be more relevant in terms of the potency we would have in patients. We noticed that depending on the electrophiles that we used, some of the electrophiles, like these aryl acrylamides, which are very common electrophiles that people have used in covalent inhibitors for cysteine, weren't performing very well in human whole blood compared to PBMCs. If we went with these alkyl acrylamides, the correlation was better. And this elicited us to do sort of this in vitro study, which was published here in this paper in JMedChem, where we actually studied the reactive, nature of various electrophilic fragments and how fast they reacted with glutathione in order to understand how reactive the electrophiles are. We thought that we would want to use electrophiles that were less reactive than very reactive ones. And sure enough, the aryl acrylamides react much more readily with glutathione than they do with these alkyl acrylamides. So reaction with glutathione, we know there's a lot of glutathione in blood. It's tied up in glutathione S transferases, particularly glutathione S transferase pi could be clearing the compound out relatively fast. So this could be the reason for the disconnect, and maybe we should focus on electrophiles in this range of how fast they react with glutathione. So this is the modeled structure of tofacitinib in JAK3. You can see the structure is modeled here. The methyl group is pointing down to cysteine 909. And one of the design concepts that we tried to execute is that we know we have an amine here leading to this amide, which actually binds to the P loop of JAK3. But this would be an easy place to install an electrophile. The problem is that, at least in this model structure of TOFA, it's pointing in the wrong direction. When we actually made those compounds, we chopped off the amide of TOFA and just put in our alkyl acrylamide electrophile. We actually did get JAK3 inhibition. This is at the km of ATP, but when we went to this 1 millimolar IC50, the potency was not very good. Here's a similar compound just without the methyl group. We weren't doing very well. Confirmation analysis, as we looked at this, pointed out what we needed to fix. So TOFA is in this series 5, where there's a methyl group here at R1. And if you look at how this bond, the energetics around how this bond rotates, TOFA is pretty much set up like this. We needed to be in this conformation in order to engage the cysteine, and the conformer energetics are pretty much the same. So it's probably up here just as much time as it's pointing down towards the cysteine. If we pulled the methyl group and replaced it with a hydrogen, now there's this electronic repulsion between the lone pair on the nitrogen of this pyrrolopyrimidine and the NH in a molecule like this, where now this conformation is favored. So we were like, that's what we need to energetically drive the electrophile towards the cysteine. And sure enough, it worked. Sometimes even in medchem, things work when you predict them. So now we had compounds that were quite potent. Our IC50s in 1 millimolar ATP were starting to be pretty good. This is actually this molecule that's sort of the fourth down is actually a locked version of this conformation. I'll show you in an X-ray structure on the next slide. And, we actually did try to even retard how reactive our electrophile would be even more than the alkyl acrylamide by putting substituents on the end, but that did not work. I think the electrophile here is just too big. So we knew we were on a good path. Again, this is the TOFA structure that I showed you. This is actually an X-ray structure of what it looks like when you clip the methyl off of TOFA and clip the methyl off of the nitrogen here. It points down and interacts with cysteine 909, and you can see it really makes this nice water bridge here between, again, the nitrogen on the pyrrolopyrimidine. There's a water that bridges to the carbonyl, and there likely is even a water bridge that goes from the nitrogen here back to this amino acid, which is pointed up towards the P loop. This is the locked conformation compound, makes the same bridge. It's energetically very favorable. We think the electrophile is pointing down towards the cysteine. There are also these aryl acrylamides. Remember on that slide a couple times back, I showed you that the aryl acrylamides really didn't perform that well, but these the energetics allows the acrylamide to point down to cysteine 909. So these worked as well. All right. In terms of thinking about how to optimize this further, remember I said we were going to need a really low dose and we were going to have to worry about clearance of these compounds. We started to notice that in order to get compounds that would perform very well in human whole blood, we were worried about the glutathione as transferase in human whole blood reacting with our molecules before it actually had a chance to react with JAK3. So we tried to think of ways in order to stabilize the compound so it would react with JAK3, not react with GST pi. And we started to introduce alkyl groups around the acrylamide to shield the acrylamide as much as we can. You had to be careful in terms of doing this. Again, more conformational analysis. You know, we actually thought that you needed to favor this conformation where the pyrrolopyrimidine is set up in an equatorial arrangement to allow the electrophile to point down. We felt that the greatest steric shielding would happen if we introduced methyl groups or larger substituents that were in an axial conformation in the cyclohexane ring. This was sort of the most energetically favored variant that we made. This is what the X-ray structure of that compound looks like. And I mean, I'm going through a lot of work that we did. We basically walked alkyl groups in all conformations around the ring in order to understand this. But this was the molecule that eventually led us to this is actually ritlecitinib, 33 nanomolar and 1 millimolar ATP. Human whole blood is 189 nanomolar IC50. Its stability in human blood was greater than, or its half-life was greater than 360 minutes. So stability in human whole blood is important in order to get sort of human whole blood IC50s down where we thought we had a chance of getting once a day dose drug. So that was actually the easy part of doing this program. The hard part, and I really have to thank the Peak PDM group at Pfizer. They did a fantastic job in working with us to sort this out. The hard part was understanding the clearance of this compound or this series of compounds. I think people have a better handle on how to do this now, but we were kind of flying blind and trying to make it up as we went along back then. So here are some studies not around TOFA, but around this compound in a locked conformation. If you take this compound, which is a JAK3 inhibitor, this analog, which doesn't have the acrylamide, it has an acetamide, so it's a model for what this compound is without the electrophile, and you start doing PK experiments. Rat PK clearance is very, very high, both for compound A and for compound B. This is at or above hepatic clearance for rat. If you do the rat PK study, but you do it in the presence of ABT, which is aminobenzyl triazole, which is a CYP inhibitor, we were hoping to tease out what was hepatic clearance based on CYP enzymes and what was conjugated clearance from GSTs primarily in the blood. And you can see this works quite well on compound B. There's no way that there can be conjugated clearance here. But even this compound, it shows you that most of this compound is being cleared by GSTs. The hepatic component is not that great, but the conjugated clearance is basically the majority of the clearance. So what we were using during the program was human whole blood stability, which has a lot of GST pi in order to optimize compounds. When we design compounds, we'd run them for potency against JAK3. Human blood stability was our surrogate in terms of trying to understand this GST pi or extra hepatic clearance. And this is actually what we did going forward. We actually noticed from some of the rat PK studies that there was this rough correlation with how reactive the cysteine is in our compounds to the in vivo clearance we were seeing in rat. The more reactive the electrophiles that we were working with, the faster the compounds being cleared, showing you again the importance of the conjugated clearance, at least in the series that we worked in. Okay, so what we eventually did, there's a lot in this slide. I'm going to refer you to the paper below, which is all of the PDM work that we did in order to understand this. So we were able to use this blood stability in order to predict the extra hepatic clearance, and we were also able to use a combination of standard hepatic scaling and empirical blood scaling in order to understand cross-species clearance, which went into how we would predict dose. Again, this IC80 is where we thought we had to be, but you need to know the potency and you need to know clearance in order to predict your dose. We calculated extrahepatic clearance by the observed blood clearance minus the scaled hepatic clearance. This plot right here kind of shows you how we were determining calculated clearance, extra hepatic clearance. We did that by noticing that there was a correlation between in vitro blood clearance and calculated extra hepatic clearance. We would primarily do this in rat, and we were able to use this calculated hepatic clearance in conjunction with the scaled hepatic clearance in order to predict in vivo clearance. First, we did it in rat. This is the one-to-one line in terms of how we predicted scale clearance and what we observed. These lines are twofold around the one-to-one. So we thought if our predictions were within twofold, we were doing okay. So that's good. And then Lewis Lung he actually had done a deep dive into the literature and found that people had actually done some extra hepatic clearance scaling by using a correlation with cardiac output. This was the formula that we used, and we were able to basically take hepatocyte clearance, blood clearance across species, and in conjugation with cardiac output, come up with what we would have as a predicted clearance across species. And we felt if we were within twofold, we were doing okay. So we first did this in rat. It's almost spot on. Mouse, sort of within twofold. Monkey, sort of within twofold. Dog lines up. Eventually, when we did the human clearance, we predicted 5.6. It turned out to be 7.3. This worked. It's completely empirical. I think if we were to do this again now, we would use basically individual GST pi enzymes and scale them directly. That also works, but at the time we didn't really have a way to do that within Pfizer. So we use this correlation in order to predict our human clearance, and fortunately it worked. Oops. Okay. So 1600 went forward into in vivo models. All of these are kind of standard autoimmune models. This is an EAE model, experimental autoimmune encephalomyelitis model. Ritlecitinib at 30 mg per kg BID, which actually is a large dose, really worked quite well. This is a 3 mg per kg dose of an S1P1 inhibitor. We also used an adjuvant-induced arthritis model where we actually induced an inflammatory response with IL-21, which actually leads to phosphorylated STAT3. You can see Ritl inhibited phospho-STAT3. It actually works quite well in the rat AIA model, and its IC50 that we had in this model lines up very, very well with the IL-21 induced phosphostat-3, 181 nanomolar. That's the same JAK3 potency we saw in human whole blood. We also looked at as in terms of occupancy with respect to STAT5 phosphorylation levels, and it also lines up roughly in a similar potency range to the JAK3 potency in human whole blood. Okay. So this compound is actually very selective across the kinome. We profiled it in a 305 kinase Invitrogen panel, a 456 kinase DiscoverX panel. Gini scores are a very high score for Invitrogen is closer you get to 1, shows you how selective again. S30, the lower the number, the better. it's a very, very selective compound. It essentially only hits kinases that were in sort of the tech kinase bin that I talked about before. Fortunately, our series didn't really hit EGFR, HER2, or HER4. It does hit some of the other JAK kinases, but we felt like even despite, the IC50s we were seeing against BTK, BMX, Tech, we had actually a fairly selective compound. In terms of trying to worry about how promiscuous this compound is, we incubated it with human serum albumin. It does not react with human serum albumin after 22 hours at room temp. Ibrutinib, which is a sort of the initial BTK inhibitor that made it to the market, does. So it shows you that it actually is pretty specific for JAK3. And then this is a way there had been work that was done around covalent labeling of hepatocytes correlating with hepatotox. So we did this type of study and came up with an estimated body burden if we were going to dose this compound at 100 mg per day, 120 mg per day, how much body burden we would get. And where these compounds, the body burden is actually higher, and these lead to hepatotoxicity, the non-hepatic toxic drugs led to less body burden. We were in that range. So we kept on doing these things to convince ourselves and the organization that we would have a compound that was safe, that wouldn't broadly label the kinome. This slide is much about, we thought we were doing a good job. This slide is really about, it's good to be lucky as well as good. So one of the things that I mentioned at the beginning of the talk is how JAK3 turns over very fast. Some of the other tech kinases do not. And this is after the compound was in the clinic and we were starting to study this, it became very clear to us that we didn't have a pure JAK3 inhibitor. We really had this JAK3 tech kinase inhibitor. Still would work very well over many autoimmune indications, but it wasn't as selective for JAK3 as we thought, again, because some of the tech kinases turn over very, very slowly. So we did these chemical biology experiments. The paper here was published in 2019, where we would dose with our JAK3 compound and actually look to compete with how well we labeled any of the kinases that our JAK3 probe would compete with, essentially. And you can see that we were getting OC50s in terms of the kinases that we labeled, where not only were we inhibiting JAK3, this is in human PBMCs, we also were labeling some of the techs. We did this in vivo experiment in NSG mice where we actually injected human PBMCs, waited 3 weeks, and then did this bolus challenge of 30 mg per kg of PF 1600, took blood draws, and actually did competition experiments with this probe. So initially, you can see across all of the techs, there wasn't much free kinase that was present. However, kinases like BTK, which turn over very, very slowly, over 24 hours, you can see BTK hasn't really come back all the way. JAK3 has come back almost all the way, as does ITK and TEC. So BTK in this study is still inhibited, and this is just on one dose. And you can imagine if you're dosing once a day, for some of the kinases that don't come back that fast, you'll continually knock them down. So not only are you inhibiting JAK3, you probably have this JAK tech inhibitor as a result of sort of multi-day dosing. So we were fortunate in that a lot of these kinases are fine to inhibit for autoimmune indications, and actually maybe there's upside to what our compound actually could do in the clinic or in how it performed. So I'll just finish with data in terms of how it worked. These are just some of the published studies. This is a phase 2 study where we studied ritlecitinib in rheumatoid arthritis. The response was very, very good. This is a sort of phase 2 study in alopecia, where we studied ritlecitinib, and also with JAK1 TYK2 inhibitor that Pfizer had developed at the same time. Ritlecitinib was safe and efficacious. This is sort of the phase 3 trial study that read out the alopecia study. Even though it's a double-blind placebo-controlled study, you can see that in cases where it worked, and it actually worked most of the time, the study unblinds itself. So that's actually quite rewarding. And you usually don't see stuff like this, but you can see the drug works. So it was approved in 2023, and importantly, I think that it was approved by the FDA for kids 12 and over. And this actually has to do with the fact that out of the JAK family, it really only hits JAK3. So the safety profile of this compound is improved compared to other treatments in this space, like JAK1 inhibition, JAK1 TYK2, or TYK2 by itself. And this compound is still in the clinic. It's in the clinic still for vitiligo, I think, which is another autoimmune disease. I think that study reads out this year. It's also in for Crohn's disease and rheumatoid arthritis, but I do not know if Pfizer plans to move that forward into registration studies. So that's really what I wanted to share today in terms of what we were thinking about, trying to take sort of a pan-JAK inhibitor, which actually was on the market or was shortly going to be on the market, and try to do something that was very specific around JAK3 in order to make this gamma chain inhibitory pathway safer inhibitor that would really be good for autoimmune indications. We lucked out a bit in terms of coming up not just with a JAK3 inhibitor, with this JAK tech inhibitor, but we actually came up with a drug that actually is quite useful and it's actually still quite safe, at least compared to some of the other JAK inhibitors. I talked to you about, this understanding of how the compound clears that actually helped us assess clearance across species and dose. And was approved in 2023, and we await how the other trials are going to read out. A very large team worked on this. My group was actually a platform medchem group that was specifically working on covalent inhibition at Pfizer from 2010 to 2015. We worked in conjunction with chemists in the inflammation and immunology research group, as well as many, many scientists across departments at Pfizer. That's what I wanted to tell you about today, and thank you very much for your attention.
Thank you. Thanks a lot, Adam. That was a fantastic story. It's all the chemistry. I mean, we've been using your, I think that 2014 paper as a reference in the group for all the GSH reactivity. Yeah, that's and all the conformational control. That's just, yeah, very amazing. I think the audience, let's just hold up your questions. You can type it in the Q&A. But then let's just go ahead. Let me introduce Dr. Minwell, and then we will have all the questions at the end. Okay. So our second speaker is Dr. Nick Minwell. He received his PhD degree from University of Sheffield under the supervision of Dr. Neville Jones, and completed the postdoc fellowship with Dr. Carl Johnson at Wayne State University. He joined the BMS in 1982, retired 2022 after having led multiple drug discovery programs, including cardiovascular, neuroscience, and virology therapeutics area. His work resulted in the advancement of 33 clinical candidates. Nick and his team was associated with the discovery of many drug candidates. This one is called MaxiPost, P3 for the treatment of stroke, H1 attachment inhibitor called fostemsavir, and HIV-1 maturation inhibitor called BMS 955176, and also the HCV NS5A inhibitor. And yeah, the list goes on. There are many, many like these, the virus antiviral drugs Nick developed. He was the recipient of the 2015 Philip Portuguese Medicinal Chemistry Lectureship Award, administered jointly by the ACS Division of Medicinal Chemistry and the Journal of Medicinal Chemistry. He was inducted into the ACS Division of Medicinal Chemistry Hall of Fame in 2015, was a co-recipient of Heroes of Chemistry Award sponsored by the American Chemical Society in 2017 and 2023, and also was a recipient of 2022 Alfred Berger Award in Medicinal Chemistry sponsored by ACS. And he was also appointed as a Fellow of American Chemical Society in August 2022. So Nick actually he wrote many not just the research papers, also reviews, and a lot of those essentially like a textbook, like in the medicinal chemistry. And so without further ado, Nick, please take over.
Well, thank you, Jim, for that kind introduction, and also for the opportunity to present here today. I've taken a slightly different approach to Adam. So, Jim asked me to talk about problems that we encountered, and so I'm going to give you vignettes from 3 different programs that all went on to be approved drugs. Let me just get rid of that. Can you guys see my slides okay?
Yes, very well.
I'll give you vignettes of the problems that we faced with each of these 3 programs. 2 of them are HCV, and I'll show you how we brought those 2 molecules together in the clinic to do a groundbreaking clinical study. Now I'm going to end up with some of the challenges we faced with developing Temsivir, actually for Stemsivir that Jim mentioned. Okay. So I'll talk initially about the protease inhibitors, which led to the discovery of a compound called asanaprovir. We took the first compound to the clinic and ran into cardiac problems, and I'll show you briefly how we fix that. For 5A, which led to discladosvir, the problem here was all preclinical. This was clinical feedback to preclinical. This was all clinical. We had a chemical issue that we had to solve to finesse the pharmacophore. And then I'll end up with a brief survey of the HIV attachment inhibitors, where, again, the problem here coming back from the clinic was solubility or dissolution limit absorption. And we fixed that with a prodrug, which I'll show you. But that created another problem because that worked so well. It unmasked flip-flop kinetics with a parent molecule, and we had to develop an extended release formulation. But it was approved in July 2020 by the FDA under the auspices of Viva Healthcare, by the way, who we transferred our virology franchise to Viva in 2015. All right, so asanaprovir and protease. This was a tough target. HCV protease. We broke our pick on this twice. I actually thought at one point it was probably not going to be inhibitable, but it was. And the compound on the left here was published in early 2000, January, by Boehringer Ingelheim. The first tripeptide base inhibitors of this protease, which has a very shallow binding groove. This was the key. This large, what we described as P2 star. It fits out. It's attached to P2 of the molecule, but it fits outside the binding groove. And this carboxylate, this is an enzyme that's subject to product inhibition. And what we wanted to do was to fill this little S1 pocket, S1 prime pocket, in this shallow groove. There weren't a lot of things to latch onto, and this one looked like it could be beneficial to us. And so what we wanted to do, we need to preserve the acidity because it's an acid. The acid product is an inhibitor, but reach through into this pocket. So the only isosteal we came up with was an acar sulfonamide shown here, and you can see that this molecule is essentially equivalent to the acid. 1 micromolar against the in cell culture and 37 nanomolar against the enzyme. But we felt we could optimize this methyl substituent, and we did that. You can see again here, here's the acid and the methyl sulfonamide. The sweet spot here is with this cyclopropyl compound shown here, where we've now got 4 nanomolar in the replicon, and by the as we got this compound, we just developed the first replicons, and a single-digit nanomolar against the enzyme. You can see that just ring expanding this, convert that to the isopropyl by adding 2 hydrogen atoms. There's a 20-fold reduction in potency for those 2 hydrogen atoms. It actually does worse than the larger ring of the cyclobutane. We've got an X-ray crystal structure, and you can see that cycloprobar group is just beautifully fitting snugly into that S1 prime pocket. And then all of the oxygen atoms around this acyl sulfonamide engage with the protein. The carbonyl group engages in the oxyanion with the NH of glycine 137, and one of the sulfone oxygens engages the backbone, the side chain of serine 139. That's the catalytic enzyme, catalytic residue. And this one, it's not shown. This one is also engaging with this glycine NH in a sort of bifurcated interaction. Hydrogen bonding interactions. That explains the potency. Now, as we continue to look at this, one observation we made, we started out with these quinoline derivatives that originated with Boehringer. We made the isoquinoline, and you can see here that that in the case of the acyl sulfonams, this applied to the acids for us as well. That the quinolines gave very poor oral exposure and liver exposure for that matter. So we looked at the chlorine here, we looked at the methoxy, and this methoxy compound, just moving that nitrogen from here down to here, dramatically affected both the plasma AUC, but also the concentration of the drug in the liver. This is at 4 hours. You can see with the methoxy compound here, we're at 64,000 nanograms per gram versus 620 for the matched molecular pair. Pretty remarkable. You can see here, it applies to the IV curve as well. You can see the quinoline is very rapidly cleared and poorly absorbed, whereas the isoquinoline is not cleared very rapidly, either IV or orally, and gives us a much better pharmacokinetic property. This molecule was the first molecule that we took into the clinic for HCV, BMS 605339. It's shown here. We did a phase 1 study, SAD and MAD in normal healthy volunteers, single ascending and multiple ascending dose. And then once we cleared safety with some of these early doses, we went into HCV infected patients. And you can see here that doses of 10, 60, and 120 within 12 hours, we've got a significant effect on viral load. At 120 mg, it's 1.8 log reduction in viremia just after a single dose. But you can see as the drug is cleared, the viral load, of course, rebounds. This looked great. This is exactly what we wanted, and this is our first experience with clinical compound. But as we looked at this more closely, we started to see cardiac adverse events. We saw mild bradycardia in some patients. We saw PR interval prolongation, junctional rhythm disturbance. And all of these, although they were asymptomatic, raised considerable concern that we'd run into somebody who would encounter this compound and have a background that would precipitate something far more significant. And, based on the exposure, which I don't show here, humans were 50 times or more sensitive to this effect than preclinical species. And we agonized over this for 2 to 3 months, trying to figure out what to do with the molecule. But in the end, we decided that we had to abandon it, and development was terminated because of these fears for significant cardiac event. So while the project development group was deliberating this in preclinical, we started working with Paul Levesque in the cardiovascular group, and he was able to reproduce the CV effects of 605339 in a rabbit Langendorf heart model. This is an isolated perfused heart in vitro. It's a beating heart taken out of the rabbit in a beaker. And we put the compound on at 10 micromolar. And this is protein free, right? So, that's a very high dose or exposure. And you can see here on the effect on heart rate over 60 minutes declines. So we'd see mild bradycardia in humans. And then over here, you see sinus node recovery time is extended by 605339. This is a very low throughput assay, and we can only screen a relatively small number of compounds. But Paul Scholar, who was the leader of the chemistry team, and Fiona McPhee, finessed out what we needed with about 20 compounds. And this was done iteratively, looking at different parts of the molecule and bringing it together with this molecule here. And you can see the only difference between these two molecules is in this P2 star. It's got a molecular weight of about 700. But we still have the methoxy, but we've moved it to C4 over here. And we've introduced a chlorine. So the only difference in the molecular formula for between these two molecules is one chlorine atom that we've added. But you can see it has a huge impact on the CV parameters. The heart rate is essentially the same as control vehicle, and so is the effect on SNRT. Again, this is at a bath concentration of 10 micromolar. So we took this molecule through advanced preclinical profiling. It was nominated for development in May of 2007 and entered clinical study in December of 2007. And that's an important date because I'll show you the next compound we took forward, which was decladazvir. These are the potency of the molecule. It's a very potent compound in both cell culture and in here and in also against the enzyme. These are the PK parameters. It had quite a different phenotype in vivo to the first compound. It was more hepatotrochic, so it distributed more effectively to the liver, which we thought actually wouldn't be a bad thing, but it turns out not to be a good thing. So the first couple of doses, 10 and 50, we really didn't see much of an effect on viral load. It's only when we went up to 200 and then 600 that we could see the effect on viral load. And again, at 200, this is about as efficacious as the previous compound comes back. And at 600, we got 2.5 log reduction, which is over 99% of reduction in viremia. Should point out the compound's named after its inventor, Li Chang-sun, so the sun comes from his name. Okay, so first molecule. Now, the second molecule, we needed, HCV is a disease that is prone to resistance, RNA virus, and we anticipated using combination therapy to avoid resistance. So once we got the first replicon assay, which was about 7 or 8 years after the discovery in HCV, and it really slowed development down. We took a chemical genetics approach, a phenotypic screen. We wanted mechanistically novel inhibitors of HCV. We used a genotype 1b replicon because that's all we had. And we developed it for HTS, high throughput screening. And I should say that this was 15 and 20 years ago. High throughput screening was 7 virologists working 6 days a week to work through this replicon. We use a dual replicon assay format in which we in the well was the HCV replicon, but we also added a BVDV replicon, bovine viral diarrhea. This is the closest phylogenetic relative of HCV. We've been using it as a surrogate for HCV. Those are in the same well. We also assessed cytotoxicity because we had orthogonal reporters. For HCV, we could look at HCV NS3 protease activity by presenting a substrate. For the BVD, we had luciferase in the genome, so we could monitor luciferase production. And then for cytotoxicity, we used Alamar blue staining. That allowed us to rapidly triage lead inhibitors based on specificity for HCV. Anything that inhibited BVDV or was cytotoxic within 10-fold of the effect on HCV, we tossed out. We initially screened about 200,000 compounds. We ultimately screened 1 million and found only one significant inhibitor. And that's the molecule shown on the right here. This had been designed as part of a prospective library by persons that I actually don't know who it was. I could never figure it out. It was a singleton. It was inactive towards NS3 protease and also to the polymerase. So it was a novel mechanism. And we could figure that out by generating resistant replicons in the presence of escalating drug concentration, map the resistant mutations, which, typically identify not only the protein target, but they also identify the binding site. And you can confirm that by reverse genetic experience, where you put those mutations back into the virus. Now, what's interesting about this molecule is the structure of it, because it's a thiazolinone with this C5 phenyl. The most typical compounds in the literature have this unsaturation here. And this had been designed, I think, for an antibacterial target. Here's the potency. It's a 0.5 micromolar in the replicon. CC50 is way above that. The BVDV inhibition is 50-fold higher. And we had BVD virus as well. There was some effect there. The effects on HIV and RSV, this is respiratory syncytial virus, are due to the cytotoxicity in those host cells. And certainly in RSV, the HEP2 cells that we used there were very sensitive to cytotoxicity. And the resistance, in this case, mapped to the HCV-NS5A protein. A Y93H change would give us at least a 10-fold increase in EC50, and this dual change pushed it out by over 100-fold. This is in domain 1, the amino terminus of a large protein that has 3 domains. And I'll show you the structure of the amino terminus. It's an essential but enigmatic HCV protein. That was the case in 2000 or in 2002 when we started this protein. It really is still the same today. It's got no non-enzymatic activity. It's now known to bind RNA, and it's now known to be involved in both replication and virion assembly. It interacts with NS3, NS4B, and S5B, and it also interacts with a range of host cell proteins. And really, even to this day, there's not a coherent picture of its function, but it's like the orchestra conductor of the virus. It controls virus replication, choreographs that, but also modulates the host cell response. So it's a fascinating target. We found out fairly quickly if we change the so first of all we looked at amino acid variation around here. We made all 20. The only one that worked was alanine and proline. Everything else was inactive. And we found out by changing this simple CBZ terminus, which is a protecting group in amino acid synthesis, to a phenylacetic acid, we get a significant increase in potency. And again, the difference between the alanine and the proline was about threefold. We did explore SAR around here. We had good dynamic range. The EC50s range from 5 to 600 nanomolar as we changed these, and it seemed to prefer more polar elements. But the SAR over here was fuzzy compared to here, because here, if you inverted this stereo center, the compound was 100-fold weaker. So very precise SAR. Now, as we continued to work with this compound, though, we ran into problems with chemical stability. We'd send a compound down to Princeton for testing. And over the weekend, it was sat in the air. So they ate the LC-MS before they did the assay and found out there was some degradation going. We figured out what that was. We could see it occurring in the replicon media. And actually what we did, we took a sample of this molecule and incubated it in media for about 3 days and then added it to the replicon. And the activity was still the same. We still had a potent EC50, even though the molecule had completely disappeared. And so we asked John Lee from the last man standing from our natural products isolation group, we recruited him. He did a biogram analysis where he took an extract from the media, put it onto an LC, pulled off different fractions and put them back on the replicon. And you can see here that, these are the various molecules. The peak of the activity was over here at fractions 61 to 62. The parent molecule, I think, is this peak here. But you can see where we're seeing the peak of activity, it's basically just a bump on the baseline. So what it did, whatever's in there was very, very, very low in concentration, a very minor component. So what we did this on a preparative scale, 100 milligrams incubated in media, and John worked out a way to extract it. And then when we looked at the LC, we could now see other peaks. We could see the parent compound. We could see these two. This is an N+16, and this is a thiourea that's derived from P2. We saw these other two peaks, peaks 4 and P6. We never did figure out P5. It's really tiny, but these two were equal. And on the replicon, you can see peak 4 was 43 nanomolar, and peak 6 was 600 picomolar. And, you can imagine that was going to pique our interest. The mass spec, John actually got enough material to do NMR on this, and he could see there was a loss of the C5 proton, and he started hypothesizing dimeric species. And when we did the mass spec, when we pushed the molecular weight out over 1,000, we could see that. And the C13 NMR showed it was a C5 to C5 dimer. And then when we heated peak 6 to 50 degrees C to homogenize the rotamers in the NMR, it actually converted to peak 4. So that's the thermodynamic stable one. That's the less thermodynamic stable compound. And this is the dimer structure. You can see we've got this C5 dimer, and that's a function of the structure of the molecule. This is a radical dimerization. So what we think happens at this site here in the parent molecule, it's very susceptible to lose a hydrogen atom, probably due to oxygen, which is a diradical in the ground state, generates a captodative radical that in the assay where it's not very soluble, it's probably aggregating, and you get some dimerization formed. We also saw hydroxylation, and that was the two products we'd seen formed in DMSO. So recall that, I said the SAR around here was good dynamic range, but fuzzy. Here it was very, very precise. So we hypothesized that this was nothing more than a scaffold to bring together a pharmacophore that is outlined in black, and this is all superfluous. And that we could test very straightforwardly because the still being a diamine, which is embedded in this molecule, is commercially available. And all you have to do is stick on this proline phenylacetic acid derivative. And we did that. We made the stilbene and we made the saturated compound. This is obviously is embedded here. This was 30 nanomolar, so that confirmed what we were hoping for. This was even more potent against the genotype 1b replicon. This was 86 picomolar. Quite a surprise. So we were pleased with this, of course. But at the time we got this data, we just developed the first genotype 1a replicon as well. And this compound was completely inactive towards genotype 1a virus, which is 60% of the infections in the US. So this had to be fixed. We also had an embedded aniline in here, which we were concerned about. This being peptide-like, could release this in vivo, release the aniline, and this particular aniline was probably going to be genotoxic based on the parent stilbene. And we're also seeing problems with this double bond with cis-trans isomerization induced by light. A couple of years after we discovered this, the crystal structure of the target was published by Tim Rice, by Tim Tellingwiesen and Charlie Rice at Rockefeller, and it showed the molecule to be a dimer. And this is just domain one. There's two more domains sitting up here. This is lined with basic amino acids, leading to the hypothesis it was an RNA binding protein that was subsequently shown. We've modeled on the amino terminus, which sits on the membrane, doesn't transverse it. These are where the resistant mutations are. So the molecule, we believe, binds across this dimer interface and interferes with the function of the molecule, of the protein. It took us about 2 years to figure out how to build in genotype 1A activity, and we had a couple of times where we thought we had some insight, but they turned out to be cul-de-sacs, and we couldn't work through them. But we ended up after quite some time identifying the cladosphere shown here. We've dealt with the stilbene problem by making this biphenyl, just getting rid of it, and then we've introduced to deal with the aniline issue. We've introduced this amide isoster, this imidazole, and the molecule is symmetrical. This is the molecule we took to the clinic. By the time we identified this molecule, we had all of the genotype replicons, and you can see it's persistently active at the 50 to 150 picomolar range. Very, very potent compound. And just before we went to the clinic, we got a genotype 2A infectious virus. The EC50 was 28 picomolar, which compares to 71 picomolar for the replicon. And again, this is the BVDV data. I'll point out, the EC50, which is for genotype 1B, in the experiment where we did this, it was about 5 picomolar. We calculated that there were 45,000 NS5As to one of our molecules. So it's outnumbered by 22,000-fold to one, which is an interesting observation. We can talk about that later. It was additive to synergistic with mechanistically distinct HCV inhibitors, 4A, NS3 inhibitors, and 5B inhibitors. So we could combine this with acinapravir, and it was safe in IND toxicology studies, and we saw no significant binding to a panel of receptors and enzyme assays. And I think Pfizer showed that there's likely an intramolecular hydrogen bond here, which explains its permeability. It does fall down on a couple of the Lipinski rules, but it's reasonably compliant. Here's the PK data. A dog was good bioavailability, monkey less so, but monkeys for us these cynos always chewed about molecules. It was taken into the clinic, and we saw a rapid reduction in viral load at doses of greater than 1 mg. There are some days at work you never forget. This was one when we saw the effects of that first milligram dose. You can see the single milligram of this compound caused almost a 1.6, 1.7 log reduction in viral load. Then over the next 4 or 5 days, slowly returned to baseline. At 10 milligrams, the viral load fell to beyond 3 logs, so 99.9% reduction in viremia. And then again, over the course of the next 5 days, it didn't really return to baseline. Then at 100 milligrams, we basically it went out to 3.5 logs and just flatlined for the whole week. This is the composition of those clinical patients, mostly genotype 1A, certainly in the 1 mg dose, or genotype 1A, and 3 to 2 in 10 and 2 to 3 in 1 mg. Because of this effect, we actually entertained the idea that we might be able to look at this as monotherapy. That turned out not to be the case, but we did explore it. We saw the rapid emergence resistance, particularly in the genotype 1A infected patients. Genotype 1A virus only requires a single mutation to produce a functional codon, where for genotype B, it requires two mutations, so it's a higher genetic barrier. Now, these two compounds, the cladribine and asanaprovir, entered the clinical study within 1 month of each other, November and December, respectively. So, having been delayed with the NS3 protease inhibitor, it went in within a month. This allowed us to do a combination clinical trial, the results of which are shown here. We had two groups. These are the genotype makeup, 9-1A in each of them, and then there were two genotype 1B in this cohort where we just gave them decladisvir and asanaprovir at the doses shown. And then the other cohort, which had just one genotype 1B patient, we gave them the two drugs along with PEG interferon ribavirin, which was the standard of care at the time. And then we looked at well, this was dosing for 24 weeks of therapy, and then we followed them for 4 and 24 weeks. 12 weeks we also monitored, and SVR is how many people were cleared of the virus at 12 weeks and then 24 weeks. At week 4, 7, and 11 were virus free. By week 24, it was 45%. The sustained virological response was actually a little bit less. We lost one of the patients here had detectable but non-quantifiable HCV at week 24, but was it undetectable on retesting 35 days later. So the number was probably actually 5 here, so a little bit higher. And you can see 90. Oh, sorry, that was one here. That was so we cured 36% of these, and we cured both genotype 1B patients and two of the genotype 1As. But over here, we basically cleared all of them. This was the first time we'd seen that two small molecules could cure HCV in the absence of immune stimulation, which redirected the entire field. And this was described as a watershed moment in the treatment of Hep C. And it set the stage for the development of the drug combinations we have today now that will cure HCV within just 2 to 3 months of therapy that has no side effects, unlike PEG interferon ribavirin. And now turn to the last vignette, which is HIV attachment. Again, a phenotypic screening lead. This is the design of the assay. It was a pseudotype virus where we've taken out the envelope proteins, we've replaced the NEF protein with a luciferase reporter, and we transfect with cells into cells that have been transfected with CCR5 to allow virus entry. It monitors for all steps of the replication pathway, integration, transcription, and translation, but it does not produce infectious virus, so we can't look at reinfection. This is earlier than the DCV screen, the decladisvir screen, 100,000 compounds again based on triage, based on therapeutic index. We found a lot of non-nucleoside reverse transcriptase, and there's a promiscuous pharmacophore. And we found a few that weren't selected for HIV, but this molecule, which came out of it, which again was from a prospective library, just a library of amides we'd bought commercially, was potent and mechanistically distinct. And this library was made up of amides and sulfonamides and was notorious for still having acyl chlorides in there. Did a lot of XAR. We couldn't change the phenyl group, maybe a thiophene, that wasn't good for us. We couldn't do much around the imidazole. We couldn't substitute the nitrogen. We could substitute with small groups at C4. We found out we could put large groups at C7. We could not substitute at C5 at all, and C6 was very poorly tolerated. The molecule chemotype, as you'll see, was blighted with solubility problems from day one. We did look at a nitrogen wart. We could install a nitrogen atom here and here and retain good activity. This was the best, and this was this. We couldn't put a nitrogen atom here. The compound was essentially inactive. We could put a nitrogen here, and it was a little bit weaker than the nitrogen here. But this was the compromise for us because it allows us to substitute over here and over here. And we knew that if we put substituents at 4 and 7, we could get an improvement in activity. This is the profile of the compound. You can see even against the panel of viruses we looked at, and T is for T cells, CXCR4. using, and M is for macrophages, CCR5 using. There's quite a range of potency, and this again was a problem with the mechanism. Even though we were targeting a conserved pocket, there was still variability, and this really was hard work for us to optimize because we needed to bring in a lot of these different viruses, and we were successful. Highly selective and not cytotoxic. This was the second molecule we took to the clinic. This is when we got clinical proof of concept for the first time that this actually worked. And again, standard PK and safety in normal health is followed by phase 2 efficacy studies in HIV-infected patients. Where here you now, instead of a single day, if they'll be able to do 7 to 14 days. In this case, we did 8 days of monotherapy. And you can see at 800 milligrams twice a day, by the time we got to day 8, we got about a 3/4 of a log reduction in viral load. This is different HCV. This is actually quite good. Tenofovir is less than a log reduction. And this compound at 1800 milligrams BID was over a log. These are the mean reductions. There was a small increase in viral load over here on day 1. That's a function of the mechanism. The problem with this molecule was that in order to get the exposure we needed to get this efficacy, this molecule had to be given with a high-fat meal to attain the target C-mean. So that's twice a day with basically a Denny's 1800 calorie breakfast, which was not going to be an acceptable requirement for development. So because of that, we recognize that we've got a problem with dissolution and solubility limit absorption, which we've been wrestling with preclinically. So what we did, we developed a phosphinoxymethyl prodrug, and this molecule is well equipped to be delivered as that kind of prodrug. Solubility of the molecules were poor, 20 mg per mil. Couldn't use. We tried formulation strategies to increase the exposure in the clinic. That didn't work. This phosphinoxymethyl group provided the solution. It's cleaved pre-systemically at the brush border membrane, so there's very little to any exposure to the prodrug in vivo. And it requires a molecule that has certain properties. It has to be highly permeable, which this was, so that it didn't precipitate out. It was absorbed as fast as it was cleaved. And the pKa of the attachment point here has to be around 9.3. Sorry, it has to be less than 13.3. This was 9.3, because if it's higher than that, the formaldehyde that's released stays on. Here's the impact of that prodrug in preclinical species, dose escalation. It's linear. It's pretty good. This compares with the effect on the capsule from the clinical trial. You can see that 200 mg capsule does not dose escalate. 200 mg solution does better. And if you feed, then you get increased exposure. And these are data in dogs. And you can see, we've got very, very poor exposure here with the clinical capsule in dogs, but when you use this prodrug, you've got this compares with the AUC from the FIH study, where again we're not getting very good exposure unless we have this high-fat meal. The optimized molecule was to install groups here that could be coplanar with the core. And this triazole, methyltriazole, and this triazole here were the 2 final molecules that we chose from. This one won out because here we had problems with CYP inhibition, and also clearance. It was higher in animal models. Temsivir is the compound shown here. It's 140 picomolar in this pseudovirus, so it's quite a bit more potent. Here's the oral bioavailability in rats, and we've got low clearance and a reasonable half-life. And we took this molecule into the clinic, both as the parent molecule, but also as the prodrug. And this is for Temsivir. And again, you can see the impact of the phosphinoxymethyl prodrug. It takes a molecule that's got a solubility of 20 micrograms per mil and increases it by over 10,000 fold to greater than 11 mg per mil. And this was taken into clinical trials as well. Here's the dose escalation for this molecule, parent molecule in plasma after dose escalation of the prodrug. Here's the clinical capsule that we use at 1800 milligrams, and you can see even at 200 milligrams we're superior. That was great. So moving along quite nicely. However, what this did was now unmask for us the problem we'd face with a parent molecule. Because of the poor solubility and the dissolution and solubility and absorption, we had flip-flop kinetics. And so what that meant was we were driving the exposure by the slow absorption. When we gave this prodrug, what we saw was rapid absorption and then rapid clearance. And so the rapid clearance was due to a mechanism of metabolic pathway that we had not seen preclinically and still do not understand, I don't believe, to this day. So what we had to do as a consequence, John Brown and Peter Timmins developed had to develop an extended release form of the drug. So what they did was they decided absorption to discern the feasibility of a slow release form of the drug. They use these IntelliSite capsules, which are marked with Indium 111 absorbed mannitol. That tracks the fate of the capsule and its contents. And we gave them the drug with watermark with technetium-99 so we could see the GI tract. And then these IntelliSite capsules have got a radio frequency activated trigger to release the drug, to pop the cap open if you like, and we release it in the proximal small intestine, the distal small intestine, and the ascending colon. We had no idea if there was if the enzyme, the phosphatase that cleave these was present there. And as you look at the profile they did calculations, computer simulations. We wanted a molecule with a low Cmax and a prolonged Cmin, and that came out of the ascending colon release time. So slow release, and that's what was developed. And that was taken into the clinic, and here are the viral load decreases. Now you can see that at day 11, after 8 days of dosing, we're seeing about a 1.5 log reduction in viral load. And these are the actual numbers done with different dosing regimens with and without ritonavir. There was a small contribution from CYP metabolism, but it wasn't significant for us here. And the molecule went on to pass phase 3. Here's the data published in this New England Journal of Medicine article in 2020. And it's now marketed by Viva's Rukobia. I'm going to stop there. I won't go through my thing. I just say that I think each program was different. Each problem was different. When we started a program, we had no idea what the pathway to a drug was going to look like, what problems we're going to encounter. And I've shown you some of the typical ones here. Some challenges are common to many programs, but our experience has been that it's usually quite different, and we have to rely upon our scientific principles I'm going to stop sharing to address these problems. And with that, I'm happy to answer any questions. And thank you again for listening.
Fantastic story. I mean, especially I think that dimer story is so fascinating, right? From a nanomolar jump to picomolar, and yeah, that was very cool. Okay, so we have about like 10 minutes, and it's open for questions. You guys can just type it in, and then I will read the questions for our speakers. So the first question is actually, I think, directly to Adam. It says like JAK-STAT pathway can be activated by many cytokines and growth factors in many cells, including hematopoietic cells. So does JAK3 inhibitor cause, I don't even know, pancytopenia? Well,
No, because it actually, so JAK3 shouldn't in terms of where it's expressed. It's really just in sort of the T cells and the NK cells. Plus, I think a lot of the warnings sort of for the heme effects of sort of JAK inhibitors potentially come from either pan-JAK inhibition or JAK2. And, I completely off the record, right? A lot of the efficacy for ritlecitinib is tech. So more so than a selective JAK3 inhibitor, that compound is probably, safer with respect to any heme effects.
I see. That's pretty cool. Actually, I have a quick question. Well, actually, two quick questions for you, Adam. So one is, I think you mentioned about this turnover rate or resistance rate of JAK3, right? So it's pretty short. There are also paper. I guess that resistance rate is determined in cell culture or in animals. Cell culture, right?
Yeah.
So
Yeah.
there are also studies actually showing, this cell culture resistance rate is not the same as, in mice or, so I wonder, because you have all these clinical studies, and so what's the half-life actually determined in, based on clinical studies objection?
Yeah, I think it's about 3.5, 4 hours the same, but you are right that I mean, we've even seen this with other covalent inhibitor programs. Sometimes when you inhibit an enzyme, its half-life changes, right? Like the cell's response is not the same. So it's a you're right in terms of, wanting to understand this more than just doing the simple pulse chase experiment, coming up with a number and thinking it's applicable across cell types. Clearly not applicable across species, right? Could be,
Yeah.
yeah.
I see. Another quick follow-up is, I mean, nowadays, these high throughput proteomics become like very popular, very powerful. So you can run, 300 samples per day. So do you think, like say, start over with this, maybe just profile at the very beginning for every covalent inhibitors using proteomics? Would that be a good strategy?
I mean, I think it depends on the opportunities that are in front of you, right? So, like, we knew that there was a cysteine in the ATP site that one could access, and we already had matter that bound there, so it was worth going for. But if you look at, I mean, Vividian's being really impressive with what they're doing with kind of a more experimental platform in order to use proteomics in order to find new leads, for a lot of the drugs they're trying to move forward. So I do like the experimental methods. I think there's, at least from what I understand, there's a bit of an art to it in terms of the size of the fragments that you screen, how reactive they are, um, what concentrations you screen at in order to get leads that actually have, a fairly selective profile to start. I think the Vividian guys, and there's actually the guys that originally started Vividian, there's another company called Expedition Med, which they're starting up now, which is, they have a lot of in-house knowledge in terms of how to do it. So
I see.
I'm completely on board, but I think there's skill in the art in terms of knowing exactly how to screen.
Yeah. Cool, thank you. So I think the next question is for Nick. Questions from a non-chemist. So what's the rationale of adding isoquinoline or quinoline moiety in the HCV drug candidates? Isoquinoline core is a rho kinase inhibitor, which plays a key role in CV function. H1152 as an example with Ki 1.6 nanomolar, which might explain its CV, I guess the cardiovascular effect, right, side effects in early development. Is a publication available for the clinical study?
With the last question, I can't remember. I would have to just go refresh my memory to see if that clinical study with the first molecule was actually published. I don't recall whether it was or not. With respect to the that P2 star element, whether it be an isoquinoline, quinoline, quinazoline, there's a lot of different things you can install there. What that does, it sits over it sits over a carboxy, an aspartate, I think it is, and an arginine, and helps them align together to help form the active site of the enzyme. This enzyme is not terribly well coordinated. It doesn't need to be a very high efficient enzyme, because if it did, if it was, it would start to interfere with the whole cell uh, integrity by cleaving host cell proteins. And, it probably does cleave a few host cell proteins, but it's not, I don't think it's a major contribution. So what that isoquinoline does, being aromatic, it sits on top of a salt bridge and stabilizes the active site. So it helps bring the active site together. So this enzyme is not very well organized, and the CMV and HSV proteases are even worse. So that's why that quinoline was there, and it was discovered initially as a naphthalene by Boehringer, and then optimized. I don't believe it's rho kinase inhibition. I don't recall that we specifically looked at that. We probably looked at a variety of things, but I can tell you that same 6-methoxy isoquinoline in a different background, in a macrocyclic protease inhibitor, was nowhere near quite the same. So there was something unique just about that peptide. And again, just because you have an isoquinone that's also an inhibitor of another enzyme class doesn't mean to say it's going to be configured in this case to interfere or bind to the rogue kinase. Um, I think you'll see a lot of different heterocycles and functionalities cross over from one inhibitory chemotype to another, different mechanisms, they're just differently configured. So I don't think we thought it was a kinase inhibition. We probably would have screened for that because we had huge kinase programs.
Yeah, cool. Thanks a lot. So this next question is for Adam. It says in cross-species clearance prediction focusing on blood and hepatocytes, could you please share why kidney clearance was not included in the analysis? Additionally, based on your experience with clearance prediction, how confident are you about extending this to predict cross-species dosing, particularly loading dose? And also, do you anticipate greater challenges in dosing prediction due to the complexity and variability of different species, different site of actions within the body? That's a
Yeah,
lot.
I don't know where to start to answer that. So I would say that the way that we did it, which was kind of jury rigged, it worked. It worked for this series. I would not say that, I would use this for, trying to do clearance predictions across species for all covalent inhibitors. I think we also had to understand exactly the clearance mechanisms of the series that we worked in. We knew that there was a hepatic component. The extra hepatic component was primarily GST pi. So that's why it worked. And I think if we were to do it today with that series, we would just, we would have access to various species GST pi enzymes, and we just scale directly from there. So, I would say for, like I'm not sure how the Vividians of the world are doing, right? Their dose predictions and clearance predictions across species. But I think when you have more than just hepatocytes, right? Or, a hepatic clearance, you need to understand how your compound is being cleared and actually try to put the assays in place that you need in order to do the predictions, both hepatic and things that are outside of the liver clearance.
Got it. Yeah. Well, actually, then just a follow-up question from another audience asking that then for covalent drug discovery, is blood stability screening always needed in addition to hepatocyte stability? I guess the answer.
I think for cysteine electrophiles, it's not a bad thing to do, and I think I've seen people do glutathione, right? Just like
Yeah.
how fast does it react with glutathione within an assay. But at least for the cysteine electrophiles that most folks use, whether it's acrylamides or Vividian's been using these vinyl sulfones, right, which react with GSH around the same amount of these alkyl acrylamides. Some sort of, how fast do they react with the cysteine nucleophile is helpful.
I see. Cool. Yeah, this question is for Nick. Given the large number of analogs generated when modifying compounds for multiple groups, what has been your most efficient approach to narrowing down the candidate list to identify those with the best PK properties, even before proceeding to in vitro or in vivo functional assay?
Well, so I haven't, yeah. So the drug discovery programs I described were from quite some time ago. We screened in vitro screens. We had to, particularly because again, I think a lot of the things we were doing were first in class, the cladosphere and fostemsavir. So we have to generate the information we need. As I was leaving Bristol Myers, retiring, we were starting to use predictive algorithms to try and anticipate and predict in vitro and in vivo properties, but it wasn't easy. You need a lot of data. In fact, I saw a presentation from somebody from Schrodinger a year ago in India, where they were really trying to do a Me Too program, a fast follow on a kinase inhibitor, I think it was. And, they did great for the first couple hundred compounds. and then have to do another 800 compounds to solve a problem, which had arisen. I think it was around Herg. So, even with countries like Schrodinger, it still remains a challenge. Typically, for us, we would make about 1,800 to 2,000 compounds to identify a clinical candidate. It just depended on the quality of that candidate. Sometimes we went forward with things that were a little bit less than ideal, because again, with something like Fostemsavir, we wanted proof of concept. The fastest one I've done was 800 compounds. That we did was 800 compounds in 2 years to get a clinical candidate. But the molecule, the template we were using only had 2 vectors to modify. And we got the very first compound we made, fixed the first structural element, one of the vectors, and that condenses the chemist onto a single place to modify. And that has a remarkable impact. When they can't go walk about all over a molecule, they end up focusing. And so there's a lesson to be learned there, I think.
Very cool, very cool. Yeah, I think we are. I think our time is up, and yeah, I really appreciate the 2 speakers sharing all these great stories. I mean, the take-home message here for all the audience: drug discovery is not easy, right? You need hard work, a lot of efforts, and like Nick mentioned, 1,800 to 2,000 compounds. That's a norm, right? So not like you can make 10 compounds and you can get something to work. So yeah, thank you, Adam. Thanks, Nick, again, and for your time, and really appreciate it. Thank you. Yeah, also
Thank you.
thank everyone for attending. Bye. Great to see you.